Okay, so this is Genetics and Molecular Biology, Lesson 3. And in this brief lesson, we are going to review DNA, the double helix, the nitrogenous bases, and how exactly does the DNA in our cell get to the point of eventually creating a protein. So let's just review the double helix first. Remember that the double helix is called the coded instructions. And DNA has the instructions for everything in our body, for everything that is made. So let's take a look at what a nucleotide is. So one nucleotide is made up of three things. We have one sugar, one phosphate, and one nitrogenous base. And that's going to be A, T, C, or G. And we've already talked about this in another unit. So this should kind of be review. Now the sugar, you might see the term somewhere, is called Deoxy Ribose. So when we look at this first thing right here, this is a nucleotide. This is a nucleotide. Here is our phosphate, evidenced by P. A is our base. And then this is our sugar. This part right here, the square where the A is attached. So you have a sugar that attaches to a phosphate group and that attaches to a base. You link all of these together and you get your string of DNA. So now we are going to move over to a different page. I need you to turn to page 24 and you should have a diagram that looks like this. It says protein synthesis, how DNA codes for the making of a protein. So the first thing, this is in the nucleus of our cell. Here's the cytoplasm, here's our nucleus. This is a string of DNA. The first step that happens, as you see here, is the DNA unzips. The second thing that happens, do you see this right here, the darker part? Okay. This is when the mRNA copies the code. So messenger RNA copies the code. And one thing that you have to remember is when this happens, A pairs with U. Okay, A pairs with U in messenger RNA. In DNA, it pairs with T, but in messenger RNA, A pairs with U. So once the messenger RNA copies the code, it leaves the cell. So mRNA leaves the nucleus. Well, where is it going now? It has the instructions. So if we're making proteins, where are proteins made? Proteins are made at the ribosome. So the next thing that is happening is the messenger RNA, here it is again, same kind of string, mRNA goes to the ribosome. Here we are. This is where protein synthesis is going to take place. Now, proteins are made at the ribosome. One part that's not really stressed on the regions, but I do want you to know about it, is the next step that's kind of hidden from us is step 5. And that's when the tRNA, transfer RNA, picks up an amino acid. Okay, so here in this diagram, this is your amino acid, AA is amino acid. And then up here, this is your tRNA. So the tRNA with the amino acid attached then goes to the ribosome. So this is where 6 happens. Okay, tRNA matches its code with mRNA. So tRNA matches up with mRNA. And that's what you see happening right here. Right here. So therefore, it's actually bringing the amino acid with it. So here's an amino acid, okay? Here's an amino acid, right here. Here's an amino acid, so it's bringing it with it. So the last thing that happens is 7, a string of amino acids make a protein. String of amino acids make a protein, okay? And that's what, so this right here is just a floating amino acid, just so you can see one by itself. And those are the steps of protein synthesis. Okay, that's it for today's lesson.